So topic 5, regulation of gene expression. Okay, so this will be from chapter 13 of your MEDA textbook. Okay, so we will cover subtopic 13.1 and 13.3 only. 13.1 is the prokaryotic regulation. Okay, 13.2 kita tak cover sebab we've already learned it before. Actually tak, just repetitive je. Okay, we don't go into detail yang eukaryotic punya regulation. So the last one is gene mutation. Okay, so what is the learning outcome for this topic? It is to describe the structure and function of an operon in the prokaryotic gene regulation. And also dalam prokaryotic gene regulation ada dua jenis operon, tapi kita akan concentrate only on the lac operon. Kita tak akan cover the TRP operon. Okay, so explain how the lac operons of prokaryotes are regulated. Okay, so this is a photo yang menunjukkan the gene expression in a prokaryote. And this is in the eukaryote macam kita belajar last in topic 4. Okay, do you see any main difference that you can tell me?
The membrane,
Okay, lagi?
the DNA,
Okay, tak juga ni, ada juga mRNA. Ni pun ada DNA dan mRNA. Ada tak lagi beza dia? One main difference between prokaryote and eukaryote that you know. The nucleus.
Cytoplasm.
Nucleus. Okay, dalam prokaryote, okay, if you can see here the cytoplasm je kan. Dia dalam prokaryote, they don't have a defined nucleus like eukaryote. Okay, eukaryote, dia ada defined nucleus. So dia ada two stages where they carry out transcription and translation. So the first part transcription occurs in the nucleus kalau dalam eukaryote. And the translation occurs outside of the nucleus, which is at the cytoplasm. Kalau prokaryote, dia satu je. Dia tak ada defined nucleus, so all occurs in the cytoplasm, transcription and also translation dia. Okay, that is the main difference between prokaryote and eukaryote. Okay, so bacteria ni dia environment memang selalu bertukar, so they do not require the same enzyme all the time. So they produce only the enzymes when they need it. Okay, dia tak ongoing. That's why they have a function operon to off and to on the regulation. So they only produce the enzymes as needed. Okay, so there was these two scientists, Jacob and Monod. They proposed the operon model to explain the regulation of the gene expression in prokaryote using E. coli. Okay, dia guna E. coli. Okay, so from dia punya experiment and everything, they proposed the operon model. What is the operon? Operon is a group of structural and regulatory genes. So when it says structural and regulatory gene ni, maksudnya you have to know dia dalam satu unit. Maksudnya transcription and the translation occurs at the same time ataupun occurs simultaneously in a single tempat. Maksudnya dalam cytoplasm sahaja. Dia tak perlu go through a post transcription and post translation punya ni, post transcription untuk nak masuk kepada cytoplasm ke apa. They don't have that. They only stay put dekat tempat the same one dalam cytoplasm. So they have a freely moved DNA lah lepas tu. Okay, so an operon is a group of structural and regulatory genes that function as a single unit. So the most important thing untuk prokaryotic regulation ni is dia punya post-transcriptional. Okay, dia punya transcriptional lagi the main point lah rather than the translation. So the main event that happens dalam prokaryotic regulation is during the post-transcription. So there are two types of E. coli operons. The first one is the lac operon, which stands for lactose. Okay, lac operon. Okay, and the last one and the other one is the tryptophan or the tryptophan. Tapi tryptophan ni kita tak cover dalam our syllabus. So we concentrate only on the lac-operon. So both operons provide typical example of an operon function. So let's take a look at what is the operon components. So dalam operon, dia mesti ada a few components that is memang functional. So first one, we have the regulator gene, which is here. Okay, regulator gene. It codes for a repressor protein controls the operon. And the repressor protein yang bawah ni, it controls whether the operon is active or not. What does it mean, the control? Maksudnya when it sticks to the operator atau when it binds to the operator, the transcription does not happen or the transcription is prevented. Okay, so that is the regulator gene and this is the repressor. Okay, the next one is the promoter. Promoter is the DNA sequence where the RNA polymerase akan attach. Okay, it's only a short segment of DNA kat sini. And the next one is the operator. So this is the binding site or the active repressor binding site dekat sini. Okay, when the repressor is active, dia akan bind to the operator to prevent the transcription from happening. Okay, the last one is the structural genes. Kalau you ada baca kat tempat-tempat lain outsource lah kan, the structural genes ni divided kepada 3. The LAC-Z, LAC-Y and LAC-A, tak silap saya. So those are the nanti ada structural genes yang akan express the enzymes lah. Dia ada 3 enzymes yang akan express dalam the lac-operon. So the structural genes yang akan transcribe, dia akan give rise to the enzyme beta-galactosidase, permease and also transacetylase. Okay. Kalau beta-galactosidase tu, they usually break down the lactose kepada glucose and galactose. Permease dia akan facilitate entry of the lactose masuk ke dalam sel. Yang the third one, yang transacetylase tu actually scientists still tak berapa nak boleh confirm what is dia punya exact function. Sebab dia macam, dia ada yang kata untuk detoxify, ada yang cakap just sebagai accessory je. So the third one yang transacetylase tu, scientists still tak dapat dia punya the function, how do you say, the specific function of it. Okay. So the structural genes, they have 1 to several genes coding for enzyme, which is 3 dekat sini of a metabolic pathway. It transcribes simultaneously as a block and it's a long segment of DNA. Okay. So this is the operon components. Okay. Lac-operon. Lactose. Okay, lac-operon. Lactose. Lactose is the sugar that can be used for food. Okay. Walaupun bacteria ni they use lactose for food, but there is not their favorite. Their favorite is glucose. They prefer kalau ada lactose and glucose, they prefer glucose rather than lactose. Why? Because lactose ni dia kena break down jadi glucose and galactose lagi. So banyak kerja. And dia rasa dia prefer they prefer glucose lagi daripada lactose. Okay, so when lactose is present in the bacteria, enzymes needed to be expressed, okay, in order to catabolize the lactose. When lactose is absent, there is no need to express the enzyme. Okay, tak ada. When lactose is absent, there is no transcription. When lactose is present, enzymes needed to be expressed. Okay. So the lac-operon is an inducible operon. The lactose, we call it as the inducer, inducible operon. So inducible operons are usually found in catabolic pathways that break down a nutrient. Okay. Alright, so kita tengok what happens when lactose is absent and lactose is present. Okay, so this photo shows when lactose is absent. Bila tak ada lactose in the cell. So when there is no lactose or any lactose sugar in the cell, the repressor, this one, the active repressor, then akan attach to the operator. Bila dia attach to the operator, RNA polymerase tak bind and the expression is off. Maksudnya tak ada transcription happen. Okay, this is when lactose is absent. Okay, so kalau tak ada lactose, you have to remember tak ada transcription happening. Okay, this is in prokaryote. Okay, so next, when lactose is present. So if lactose is present, it combines with the repressor. Okay, so lactose berada kat luar sel ni, kat dalam sel, sorry. Dia akan combine dengan the active repressor. Bila dia combine, it makes the repressor inactive because dia dah change its shape. Saya ambil kawan awak masa ni. Lama. Macam nak tekan? Okay, alright. So active repressor, if there's lactose around, lactose will bind to the active repressor, making it inactive by changing its shape. Okay, bila dia dah tukar shape, dia tak boleh bind to the operator. So this is the operator. Okay, bila dia dah tak boleh bind, dia tak boleh stop the transcription. So the RNA polymerase can now bind to the promoter. Okay, so when it binds to the promoter, then transcription can happen. So the 3 enzymes are then synthesized, which is kat bahagian the structural genes, dekat the this long segment of DNA. Okay, dia akan transcribe semua ni. So akan keluar 3 enzyme, which is beta-galactosidase, permease, and also transacetylase. Okay, that is the difference between lactose is present and lactose is absent only. Maksudnya tak ada, there's no other sugar like glucose. Masa lactose sahaja. Okay, jom kita tengok video. Ya.
Maksudnya the main purpose for lac operon punya sistem ni untuk produce 3 enzim tu.
Okay, actually to produce, yeah, betul lah. To produce, okay, dalam cell, it needs food, right? It needs sugar. Bacteria also. So yang lac operon ni function dia actually as a backup. Kalau ada glucose, the bacteria akan guna glucose tu instead of the lactose. So lactose ni katakanlah kalau dalam sel bacteria tu dah tak ada dah glucose, only then the lac operon akan operate. They serve as a backup untuk survival. Okay, bila dia dah bila dia dah release the 3 enzyme tu, which is beta-galactosidase, permease, and then lagi satu transacetylase. Tapi the main dia ialah beta-galactosidase. Yang beta-galactosidase enzyme ni yang akan pecahkan, break down the lactose balik kepada glucose and galactose. So then ada glucose balik dalam badan bakteria tu. Okay, that is the function of beta-galactosidase. Yang that has been transcribed to, and then it goes through synthesis, hasilkan beta-galactosidase yang akan pecahkan atau break down the lactose into galactose and glucose. So the bacteria needs glucose. Okay, yang permease ni pula function dia untuk bawa masuk banyak-banyak lactose dalam sel. Dia akan bind to the membrane. Dia ada lubang ni kalau kita tengok. Okay. So, jom tengok the video. I think it's better you understand lagi lah if you watch the video.
Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Yang tu bila only the lactose is present, maksudnya dah tak ada glucose dalam sel. Okay, now we will go on into what happen kalau ada glucose dalam sel. So what happen to the operon, sama ada dia function ke tak kalau ada glucose. So you can see the first sentence here, the E. coli preferentially breaks down glucose dari compared to the lactose. Sebab glucose ni is already a simpler sugar compared to the lactose. Lactose nak kena break down into glucose dan galactose lagi. So, the lac operon is maximally activated only in the absence of glucose. Bila tak ada glucose, baru dia maximally operating. Okay? So, the presence of glucose will inhibit the catabolism of lactose. Okay? So, dalam glucose ini is a bit more complex sikit compared to the kalau lactose sahaja yang present, which involves the repressor. Kalau okay, senang kalau you nak ingat repressor tu, you ingat lactose. Okay, only lactose saja. Kalau CAP, CAP ni, this one here, catabolite activated protein, is like a glucose signal. Okay, kalau dalam lactose tak ada CAP, only kalau dalam lactose dia tak ada CAP, dia only ada repressor. Kalau when there's like the glucose around, this will be activated, CAP. Okay, so when glucose is absent, there is a signal, CAMP. We call that as a hunger signal ataupun starvation signal. Maksudnya dia tengah lapar bila tak ada glucose kan. When glucose is absent, the CAMP, okay, dia akan attach to the CAP, catabolite activated protein, dia akan jadi CAMP-CAP complex. Okay, this complex akan attach to the binding site, CAP binding site, tepi promoter, next to the lac promoter. Okay, so kita tengok dia punya ni. Okay, this is a bit different if you notice. Okay, this is the cap binding site. Tadi mula-mula yang dalam yang lactose availability sahaja, dia tak ada cap binding site dekat sini. Dia ada promoter and dia ada operator. Okay, for this one, when glucose is absent and lactose is present, okay, ada lactose tapi tak ada glucose. So, the CAMP accumulate. Okay, dia dah lapar. So, this is the starvation signal. So, that means bila dia accumulate banyak-banyak, the CAMP will be high in the cell. So, bila CAMP is high, it binds to the CAP. This one here, making it active. Mula-mula CAP ni inactive. But when there's a build-up of CAMP, dia akan bind to the inactive CAP, we will make it active. Okay, when it is active, this cap akan bind to the cap binding site dekat tepi promoter. Okay, when the cap is bound, the RNA polymerase, it binds better to the promoter. Dia penuh. You can see here gambar dia penuh. So the RNA polymerase binds fully with the promoter. So lastly akan ada transcription happening because the RNA polymerase boleh bind to the promoter. So the structural genes of the lac operon are expressed more efficiently. Okay, so this is when glucose is absent. Bila glucose is absent, you have to remember the CAMP bind to the CAP. Kalau dalam lac tadi tu, lactose, dia cuma ada repressor dekat operator. Sama ada turn on or turn off. Kalau glucose is absent, it involves CAMP and CAP complex. Dia akan bind to the cap binding site. Okay? Okay, so what happens when glucose is present? So this is glucose is absent and lactose is present. So now we'll see when glucose is present. What happened? Okay, when glucose is present, there is little CAMP in the cell. Okay, there is no accumulation sebab when glucose is present, they prefer to use the glucose daripada lactose. So, bila dia prefer to use the glucose, there is little CAMP inside the cell, so the cap will become inactive atau stays inactive. Okay, this one here. Tak ada. So, the inactive cap does not bind to the CAP binding site. So, bila dia tak bind, this RNA polymerase, it does not bind fully. Kalau you tengok, dia bind separuh je. Okay? The RNA polymerase does not bind fully with the promoter. So, lactose won't be transcripted, the structural genes won't be transcripted and the lactose won't be catabolized. Okay? So, that is when glucose is present. Okay, so this is the summary of the lac operon responses. Sekejap ya, saya nak cakap anak saya tengah bising kejap. Kairash, mama tengah kelas. Sekejap, okay? Okay, alright. So this is the summary of the lac operon responses. Kita boleh tengok there are 4 responses altogether. There's glucose present and lactose absent. Glucose present and lactose present. Glucose absent and lactose absent. Glucose absent and lactose is present. So kalau you nak tengok, senang dekat jadual dekat bawah ni. Okay, so when there is presence of glucose and lactose is absent, there is no transcription. Gambar ni. Okay, the glucose is present, lactose is absent. So when glucose is present, there is no CAMP, accumulation of CAMP. So dia tak bind to the cap and this cap tak bind to the cap binding site. Okay, so when glucose is present and lactose is present, dua-dua is present, what happened? So, walaupun when glucose is present, you have to link it dengan CAMP and CAP. So, bila glucose present, tak ada. It doesn't bind to the cap site. However, because lactose is present, dia boleh, the repressor tak bind to the operator. So, RNA polymerase still boleh berjalan dekat sini. Still, there is transcription, but it is low level. Low level of transcription. Okay, because the repressor tak bind to the operator. Okay. Next one. When glucose is absent and lactose is absent. So, bila you tengok glucose absent saja, you dah boleh link dengan CAMP and CAP. So, dia akan bind to the cap site. Okay, cuma when lactose is absent, akan ada repressor. Repressor akan bind to the operator. So, still tak ada transcription. Okay, kalau you perasan pattern dia dekat sini, repressor sini repressor bind to the operator because the lactose is absent. Okay, so last one, when glucose is absent and lactose is present. So glucose absent, you have the binding site dekat cap ni. And then the lactose also present. So there is transcription, maksudnya repressor tak bind to the operator. So bila dua-dua ni bergabung, glucose absent and lactose is present, there is a strong transcription happening. So there's high level of transcription. Okay, so you have to remember this summary of lac operon. Maksudnya respons apa kalau ada, macam dalam exam dia akan bagi situation. And then you have to analyze the situation, glucose present ke, lactose absent ke, apa-apa. So please describe based on what you understand when there is glucose present and ataupun when there is the absence of glucose, absence of lactose or presence of lactose. Okay? Alright, so next we go on. Yeah.
Apa function sebenarnya untuk CAP ni?
Oh, tak. CAP ni dia akan make the promoter to fully bind. Yang ni. Ni. Kalau you compare tadi gambar tadi. Bila CAP ni dia bind to the binding site, dia punya apa ni? Dia macam the RNA polymerase boleh bind fully. Kalau dia tak ada, you tengok RNA tak boleh bind fully. Dia macam promote the thing lagi. Promote the binding of the RNA polymerase. Kalau bila dia dah bind separuh ni, dia tak jalan. Kalau yang gambar tadi, kan bila dia bind, dia RNA bind fully to the promoter. Nampak dia penuh je dekat promoter tu. So dia boleh berjalan. Dia boleh jalan dekat the DNA ni. Okay, dia boleh transcribe dah. Kalau dia separuh je, dia tak boleh. Dia tak boleh nak transcribe. Dia kena penuh. Okay. That's why we need the help of the cap binding site. Okay, when you mention about the cap ni, okay, we will see about the negative control and the positive control. Kalau you tengok tadi, we need the cap tu kan untuk nak enhance lagi the transcription. So So the cap tu is actually a contoh of a positive control. They enhance lagi the factor. Okay, kalau negative control, contoh macam repressor. Okay, in negative control, an active repressor, bila repressor is active, it blocks the gene expression by binding to an operator site. Kan? Kalau dia aktif, dia bind kepada operator, there is no transcription. Terbalik pula kan? Active, no transcription. So this is negative control. When it is inactive, it allows gene expression. Sebab when it is inactive, dia tak bind to the operator and transcription happens. Okay, this is negative control. Okay, kalau positive control, active factor is required for gene expression. So the small molecules can convert an inactive factor into an active one. Okay, kalau inactive positive control, factor pula result in no gene expression. Inactive, no gene expression. When there's active factor, ada transcription. Okay, this is example of negative control, yang ni positive control. Okay, so kalau you kena ingatlah kalau negative control tu selalunya deals dengan repressor kalau dalam lac operon. Kalau positive control tu is the CAMP and CAP. Okay. Right, so that is all for prokaryotic regulation. Ada soalan tak on prokaryotic regulation? Dia beza kan? So that means bila dia dah transcribe dekat sini, ingat tak yang saya cakap the main event of the prokaryotic regulation is the transcription. So bila dia dah dapat mRNA from the transcription, baru dia akan translate into the enzyme yang 3 tadi tu. Okay, that's why I say it happens in one place. Only in the cytoplasm. Dia tak payah nak go through post transcription, splicing semua tu dalam nukleus dulu baru keluar kepada cytoplasm. That one is only in eukaryote. So this is prokaryote. Okay? So that is the main difference between eukaryote and prokaryote punya gene regulation. Okay, now we are done with prokaryotic and also kita dah tahu dah eukaryotic macam mana. So next we go on into gene mutation. Okay, what is gene mutation? It's the permanent change in the sequence of bases in the DNA. Okay, sometimes what are the effects? Sometimes dia akan ada no effect and sometimes it can have the complete inactivation of the protein. Dia boleh vary dua, daripada teruk ataupun ke no effect langsung. Okay, there's two types of mutation dekat sini. Spontaneous and induced. So when people say germline mutation, you have to refer it to sex cells. Okay, gametes. Kalau somatic mutation, you link it to the body cells ataupun body cells mutation. Okay, this is the gametes. Okay, so two causes of the mutation. It can happen spontaneously and it can be induced mutation. Okay, apa beza dia? Spontaneous and induced. Kalau spontaneous mutation, dia selalunya melibatkan mispairing during replication. Okay, it happens naturally in our body, so we call it as spontaneous mutation. ataupun the movement of transposon from one chromosomal location to another. Maksudnya there's a base substitution daripada satu point kepada satu point yang lain, ataupun replication error. Tapi error punya part ni selalunya we don't take it something seriously because we have the DNA polymerase. DNA polymerase ni dia sangat-sangat efficient. Dia boleh detect the overall mutation rate ni satu dalam satu berapa ni? Dah tak tahu dah saya. Okay? So because DNA polymerase is very efficient that it can proofread all the new strand and correct the errors. So selalunya when we say spontaneous mutation, selalunya lah kita akan relate it to the mispairing during replication. Sama ada during replication pun atau S phase replication ataupun during the mitosis phase itself lah. Okay. Another one is induced mutation. Induce, it needs something yang trigger it. So apa yang trigger? It can be caused by mutagen. Mutagen ni contohnya macam radiation and also chemicals. So many mutagens are also carcinogen. Carcinogenic, okay, it means that it is cancer causing. So kalau kita tengok dalam environment kita, the mutagens are usually ultraviolet radiation, UV, smoking and X-ray. Tapi ada banyak lagi all the other factors yang can lead to induced mutation. Okay. Selalunya kalau dalam exam, dia akan bagi situation ke macam mana, and then you analyze benda tu sama ada it is spontaneous mutation ke induced mutation, ataupun dia suruh bagi contoh induced mutation macam mana. Okay. Alright. So sekarang scientists what they do untuk nak detect mutagens atau the cancer causing punya chemical, they use the Ames test. The Ames test. Okay, yang ni what you have to know, you don't have to know in detail, cuma yang ni just to give you the overall view macam mana diorang buat the experiment. So kalau dia nak test a certain chemical tu sama ada carcinogenic or not, so dia akan masukkan the suspected chemical mutagen dalam ni dalam a bacterial strain. Bacterial strain ni dia perlukan histidine, which is the essential amino acid untuk growth. Okay, kalau you tengok dalam ni, dia letak dalam control is just the bacterial strain sahaja without the chemical, the suspected chemical yang diorang nak test tu. Okay, so then they put it on the Petri plate, Petri dish. Okay, dia biar overnight. Kalau you tengok dia punya result ni, there's a bacterial growth untuk the mutagen. Kalau normal punya ni, which requires histidine, bacterial strain requires histidine, tapi atas plate ni dia tak ada histidine, dia tak grow. Mutation does not occur. But in here, kalau you tengok, kalau the mutagen, the chemical tu is carcinogenic, dia mutation akan occur. So there is still growth of the bacterial cell. So that is how scientists akan check whether a certain chemical, the pesticide ke insecticide ni is carcinogenic or not. Okay, they do the Ames test. Okay, next we go on into the effect of mutation on protein activity. Okay, mutation ni sebenarnya ada dua type. Point mutation and also frame shift mutation. So when we say point mutation, it means that satu je, one type of point mutation is a base substitution, kan? Maksudnya dia kacau satu je, one single. Contoh yang dekat sini. So this is base substitution. Dia tak involve the whole thing. Kalau dia kacau pun dia kacau satu je amino acid dekat sini. Dia tak kacau yang dekat belakang-belakang. Okay, this is called point mutation. They change only one codon to a different codon. It involves a change in only a single DNA nucleotide. Okay? Single DNA nucleotide. Kalau you tengok sini normal, ni sepatutnya A. Tapi bila there's a base substitution, dia akan masukkan G. So bila dia masukkan G, dia akan complement dengan C. So dia akan hasilkan another amino acid which is different. So dia akan carry out another function daripada function asal dia. Okay? So that is point mutation. Okay, point mutation ni selalunya dia akan lead to non-functional dia akan reduce sama ada non terus, non-functional ataupun reduce functionality ataupun it can be unaffected. Okay. Okay, next. Okay, ni contoh point mutation. We have the sickle shaped blood, sickle cell anemia tu kan. Kalau when the sickle cell anemia, dia ada change in the DNA dekat sini, dekat mana ni? Sini, ya. The glutamine. The glutamine tu akan jadi valine. Okay, so dekat apa ni? T ni dia tukar satu je kan. When it tukar satu saja, you know that it is a point mutation. When point mutation, dia akan complement and then dia akan jadi a different amino acid. Okay, kita tengok kat sini. So this leads to the, kalau glutamine ni dia akan jadi the normal shape. Kalau valine dia tak akan jadi retain the original shape. Dia akan jadi sickle shape. So the absorption of the blood makin, makin berkurangan because of the lower surface area. Okay. So that causes anemia. Okay, next one. Macam saya cakap tadi, ada dua point and frame shift. So this is frame shift. So what happened during frame shift is whether one or two nucleotides are either inserted or deleted from the DNA. Okay, this is a normal one. Okay, saya bagi contoh mula-mula. So the protein selalunya bila yang dah mutated tu, dia tak akan function. Dia akan jadi non-functional. Contoh kalau normal kita, the cat ate the rat. Okay. Kalau after deletion, contohnya kita buang C. Okay. The Ata ate her ate. Dah tak ada kan? Jadi dia dah ubah the whole sampai dekat belakang ni. Dia bukan ubah satu je. Bila this is what it caused by frame shift mutation. Kalau tadi dia ubah satu nucleotide. Tapi yang ni dia ubah satu atau dua but by deleting or inserting. Okay, so yang ni dia delete C ni. So yang belakang-belakang ni semua berubah. So that's why it's called frame shift mutation. Katakanlah kalau insertion pula. Dia tambah another C dekat yang cat ni. Dekat depan cat. Another C. So dia dah jadi pun lain. The, ka, tat, ra, era, tat. Okay. So dia dah jadi nonsense lah lepas tu. So this is frame shift mutation. Okay, contoh kalau dalam gambar, addition here, dia akan ubah contohnya daripada sini, you tengok the amino acid, the whole amino acid kat belakang semua akan berubah. So this is frame shift mutation. Belakang pun sama, kalau yang ni pun sama, kalau deletion, okay, T satu keluar. So akan ada less one amino acid which can change the function ataupun specific function of protein. Okay, so that is frame shift mutation. Okay, yang tadi saya dah sebut non-functional. What happen when it becomes non-functional protein? Okay, you have to note that a single satu je non-functioning protein can have a dramatic effect on the phenotype. Okay, so if a faulty enzyme is inserted into a metabolic pathway, a person may be unable to convert one molecule to another with serious consequences. Okay, serious consequences sama ada frame shift mutation lah. Okay, for example, kalau kita tengok dekat bawah ni. The example here, which is on page 240. Okay. For this, the first one, the disorder phenylketonuria, PKU, is when the phenylalanine builds up in the system and the excess causes an intellectual disability. So it is the build up of the phenylalanine. Sebab bila enzim A ni malfunction, dia unable to convert A kepada B. So bila dia unable to convert, yang ni akan jadi in abundance. Bila dia build up banyak-banyak, dia akan lead to disorders. So dalam phenylalanine punya case ni, dia akan lead to phenylketonuria, which is intellectual disability. Okay, another contoh, kalau tyrosine ni, okay, dia ada consequences. So yang ni tak boleh nak convert pada yang ni pula tak boleh nak convert. So katakanlah kalau jadi kat sini, habis semua tak boleh convert. Kalau jadi kat sini, then yang ni tak ada effect, yang ni akan convert. Okay, contoh kalau enzim B pula malfunction, is unable to convert this tyrosine kepada melanin. So that person akan ada albino. Okay, so these are the events yang dia akan cause serious consequences when there is a single non-functional protein. Okay, alright. So lastly is the mutations that can cause cancer. So the remember as I said before, cancer causing punya ni they rely on dua genes which is proto oncogene ataupun oncogene bila dia jadi cancer causing cell. The another one is the tumor suppressor gene. Okay, kalau oncogene ni dia stimulate cell division. Kalau tumor suppressor, kalau yang ni pun dia punya keyword dah. Tumor suppressor gene ni dia inhibit the cell division. If there is any mutation towards this genes, dia akan replicate, dia akan jadi banyak. Contohnya kalau oncogene ni, mutated proto-oncogenes becomes oncogene, which is the cause cancer. Kalau oncogene tu you boleh tahu dah, it has the potential to cause cancer. And because this oncogene, dia boleh always active. Dia akan keep on replicating and keep on replicating. Tu yang jadi banyak and then jadi cancer. So another one, kalau tumor suppressor gene ni selalunya gene P53. Dia inhibit the cell division sepatutnya. Tapi bila there's a mutation happen dekat tumor suppressor gene ni, dia stimulate the cell cycle uncontrollably. Dia tak suppress dah. Dia tak inhibit. So this can lead to tumor formation. Okay, so these two genes yang usually involved with cancer. They are the cancer causing agents. Okay, oncogene and tumor suppressor gene. Okay, yang ni this one you don't have to know, ni saja saya letak just for your own info lah. Okay, so bila ada contohnya kalau yang ni P53 gene, kalau tumor suppressor gene, so dia akan kacau dekat bahagian transcription because it is a transcription factor. Dia akan kacau dekat transcription which makes the protein that is unable to inhibit the cell cycle ataupun dia tak boleh promote apoptosis. So dia akan keep on replicating. Okay. So next one is the oncogene, sama juga. Dia jadi transcription factor. So dia akan kacau dekat bahagian transcription and then the protein produce proteins that overstimulate the cell cycle. Dia akan keep on replicating.